Good morning, everyone. I'm Scott Weinberger, and I am the moderator today for the webinar being presented jointly by GenX Technologies and Neoproteomics entitled Hydroxyl Radical Protein Footprinting Data Processing in a Higher Order Structural Analysis, as being presented by Professor Joshua Sharp of the University of Mississippi. Today's seminar is a part of a 5-segment program that has been co-produced by GenX Technologies and Neoproteomics. Our two companies have come together to create an alliance in protein footprinting with the underpinning desire to increase the overall prevalence and utility and awareness of protein footprinting technology and products and services in the biopharmaceutical arena. So we formed this alliance to facilitate the rapid exchange of scholarly content and discourse among researchers such as yourselves working in pharmaceutical and biopharmaceutical development and structural biological studies. Neoproteomics is a world-recognized leader in footprinting services for biopharmaceutical and structural biological research, and GenX Technologies is the world's only provider of footprinting instruments and specific software and accessories that enable higher-order structural analysis to be performed in a compact, intuitively simple, safe, and benchtop format. As I mentioned moments ago, this is part of a 5-component series. The first 3 sessions, which have already been produced, an introduction to protein footprinting applications in structural biology, and fast photochemical oxidation of proteins, are currently available on demand at our website at genxtech.com. Of course, today's session is going to be provided live by Professor Sharp, and it will ultimately, after its production within a few working days, also be available for on-demand viewing. And the final installment of this series will be presented by Professor Lisa Jones at the University of Maryland next year at February 10th at 11 a.m. Pacific Standard Time. And Lisa will basically review her work and her revolutionary advancements for in-cell and in vivo FPOP. And now a few words about today's presenter, Professor Joshua Sharp of the University of Mississippi, and also Chief Technical Officer for GenX Technologies Incorporated. Professor Sharp is a world-renowned and internationally recognized expert in hydroxyl radical protein footprinting. He has published more than 25 peer-reviewed articles in the field of HRPF, methods development, fundamentals, and applications between 2002 and today, with over 45 publications overall. Professor Sharp published the first benchtop methods for hydroxyl radical protein footprinting, including the first method using UV photolysis of hydrogen peroxide for hydroxyl radical generation. With numerous invited lectures to both academia and private industry on the applications of HRPF to biotherapeutics, Dr. Sharp has addressed problems associated with formulation, biosimilarity, aggregation, and protein-protein and protein-ligand interaction mapping. Professor Sharp's laboratory is also known for their work in glycosaminoglycan structure function analysis, and his team commonly leverages their expertise in both areas to study protein-carbohydrate complexes. Professor Sharp received his PhD in 2003 from a joint program between Oak Ridge National Laboratory and the University of Tennessee under the mentorship of Dr. Robert Hedich. He performed his postdoctoral studies at the National Institute of Environmental Health Sciences in the Laboratory of Structural Biology under the supervision of Dr. Kenneth Tomer. In 2007, he joined the University of Georgia's Complex Carbohydrate Research Center as a research faculty member. Most recently, Professor Sharp joined the faculty at the University of Mississippi School of Pharmacy, where he currently serves as an associate professor of pharmacology and a research professor of the Research Institute of Pharmaceutical Sciences. He has received multiple awards for excellence in research, most recently winning the New Investigator Research Award from the University of Mississippi School of Pharmacy and being designated the University of Mississippi nominee for the 2019 Blavatnik Award in Life Sciences. Professor Sharp serves on multiple national and international advisory and review panels as an expert on protein and carbohydrate biophysical and structural analysis by mass spectrometry. It is now my sincere honor to introduce to you all Professor Joshua Sharp. Josh, it's all yours.
Okay, thank you very much, Scott, for that introduction. Okay, so hopefully now everyone can see my screen. So again, my name is Joshua Sharp at the University of Mississippi. I've been doing hydroxyl radical protein footprinting for a very long time. And today I am going to talk to you a bit about data analysis, how we actually analyze the data, which is one of the most common questions that we get from people that are wanting to get into hydroxyl radical protein footprinting. Is the data seem really complicated? How do you actually sit down and analyze it? So I'm going to go over that today, give you some examples of the kinds of data that we normally see, how we go about interpreting that manually, and then I'm going to talk a little bit about software solutions that are currently available. Before I get started, while my day job is professor at the University of Mississippi, I do have a financial conflict of interest. I have a significant financial stake in GenX Technologies, and I will be talking about software that is being offered by GenX, so please keep that in mind. Okay, so as Scott mentioned before, this is part 4 of a 5-part series. I am going to go into this assuming that you have some idea about what hydroxyl radical protein footprinting is, how it works, what you can use it for, what kinds of information it's supposed to give you. We're really going to focus on how to hands-on analyze and interpret HRPF data. If you feel like you don't have a good grasp about kind of the fundamentals of what HRPF and FPOP are, I strongly encourage you to take a look at some of these previous on-demand videos that we have from this series. I personally am very proud of them, even the ones I didn't do myself. I think they're an excellent introduction to the field. So just so we can all get oriented about the experiment that we're talking about here, I'm going to give you an idea of the general hydroxyl radical protein footprinting workflow. Typically what you have is you have a protein in 2 different structural conditions. So here I've got a protein ligand bound versus ligand free, but it could be formulation A versus formulation B. In anything where you have suspected structural differences between the same protein in 2 different conditions. You expose the protein to hydroxyl radicals diffusing in solution. They oxidize amino acid side chains that they encounter on the surface of the protein, and they form stable protein oxidation products. These protein oxidation products, the rate at which they're formed in any particular area depends on the identity of the amino acid, but also how exposed that amino acid is to the solvent. So when things happen to the structure, for example, a ligand binds and shields a particular region, then you could go from strong oxidation in the absence of the ligand to poor oxidation in the presence of the ligand. So you'll see that change in the surface of the protein based on the pattern of oxidations. These oxidation products are stable, so now you can denature and digest your protein, use your favorite protein workup protocol, and you end up with a pattern of oxidized peptides, where the pattern of oxidations that was present in the original protein are preserved in the pattern of oxidation on your peptides. You can measure this oxidation using LCMS, where you can identify both the original peptide and the various oxidation products. You can relatively quantify these relative oxidation products, and you can use that quantitative measurement in order to determine a relative amount of oxidation of each peptide in condition A versus condition B and see how that changes. And then based on that changes, based on those changes in oxidation, you can go in and measure changes in protein topography or surface. What parts of the surface changed when you made that change from structural condition A to structural condition B. Some places became less oxidized and therefore less exposed to solvent. Some places may have become more exposed to solvent. So that's just a general idea of hydroxyl radical protein footprinting and the workflow. We're going to focus on this part of the workflow here. So we've gotten our oxidized peptides, we've done the LCMS, and now we have the data. How do we take the data and turn it into something like this that we can interpret and generate structural information out of? And so we're going to focus on what's known as peptide level quantification of HRPF oxidation, where we're looking at oxidation at the level of the peptides that you generate when you digest your protein. Okay, we're going to go through the process step by step and talk about each step, why we do that step, why it's important, what some of the common pitfalls are as we go through this process. So the 3 basic steps in doing peptide level quantification. The first step is you want to identify all of your unoxidized peptides that you can identify from your protein. This kind of gives you a base level of how your protein digested, what parts of the protein you can see, and what those unmodified peptides normally look at. The next step is we search for major oxidation products of each identified peptide at the charge state in which we originally identified that peptide. And there are various major oxidation products, most of which have been pretty thoroughly characterized over the years. There's the net addition of one or more oxygen atoms. There's loss of formaldehyde from aspartic and glutamic acids. There's a deguanidination reaction for arginine. There's carbonyl formation from lots of amino acids. It's usually a minor product, but a lot of times you can detect it. And then there are a variety of histidine modification products that result from the opening of the imidazole ring. And these are also usually minor. Once you've searched for all of your major oxidation products for every peptide, then you go in and you quantify the unoxidized peptides, and you quantify all of the detected oxidation products based on their intensity in the mass spectrum. And there are a few tricks that you use to do this, and I'll go over those tricks and why they're important as we go through this step by step. So the first step that we always take when we're doing this quantitation is you want to find your unoxidized peptides. And fortunately, the tools for doing this are very robust nowadays. Compared to a lot of high throughput shotgun proteomics, most HRPF experiments are usually relatively simple. Usually you're looking at a single protein or maybe a fairly simple mixture of proteins. And so if you're just looking at the LCMS profile of the unoxidized sample, it's pretty simple. You're going to see peptides from that one protein or that small collection of proteins. It's not usually going to be a terribly complex mixture. So most of your basic software does a really good job of this. But it's not necessarily as simple as you might think. So in addition to the protein that your analyte protein, you also have to keep in mind you've added in catalase as part of your quenching process. You've added in some kind of protease that's going to have some autodigestion products. But in addition to that, you're going to have all of the protein oxidation products. And even though those protein oxidation products are not what you're looking at right now, they can actually cause some interference if you're trying to look at, if you're trying to identify unoxidized peptides just based on something like their mass. And as an example, what I've shown here is I've shown a mass to charge of an unoxidized peptide. I've made an extracted ion chromatogram looking at the mass to charge of an unoxidized peptide that I know exists in this mixture with a mass error of 10 parts per million. And what you can see is, while this is a single protein, a single analyte protein, you can see I've got three different peaks, three different clear peaks that show up within this 10 ppm error. And that's because, remember, in addition to all of the peptides I have for my analyte protein, I have the trypsin, I have the catalase, and I have the oxidation products of a lot of these different peptides, all of which could have overlapping mass with the mass of the unoxidized peptides I'm looking for. So when you're trying to identify your unoxidized peptides, you generally want decent MSMS spectra so that you can get confident identification of your unmodified or your unoxidized peptide. And once you have that confident identification of your unoxidized peptide, that gives you information that's really going to be key to use as you're trying to identify your oxidation products. But fortunately, that step is pretty straightforward. Unoxidized peptides tend to be more abundant in the spectrum. And again, the computational tools we have for identifying unmodified peptides from LCM SMS data are very robust, very well understood. So I'm not going to belabor that point too much. I'm going to move on to step 2, which is the first step that's really somewhat unique to hydroxyl radical protein footprinting. And that is making basically a theoretical list of your potential major oxidation products for each peptide, and then searching for those major oxidation products. And in order to limit what you're going to look at, we're going to use a little bit of expert knowledge to limit this. And this is where the identification of your unmodified peptide comes into play. So there are lots of different modifications that are possible. Okay, the most common ones and the ones that are most commonly reported are the addition of one or more oxygen atoms. So plus 15.995 Daltons, plus 31.990, et cetera. But loss of formaldehyde from acidic amino acids is also very common for peptides that contain acidic amino acids. The addition of carbonyls, so addition of oxygen and loss of H2, so the addition of about 14 Daltons is relatively, you see it, it's not unusual. And so there are lots of different oxidation products you can look for. And if you try to look for every oxidation product of every possible peptide from a theoretical digest at every possible charge state, very quickly the different combinations become unmanageable. It's just way too much to look at. But what we know is, with one very notable exception, in positive ion mode, these oxidation products tend not to change the charge state of the peptide where the modification occurs. So we know when we identify a given unmodified peptide in a +2 charge state, we know that we can look for the oxidation products in that same +2 charge state and still still be confident that if those oxidation products exist, we will see them. The one notable exception that I'll mention is this deguanidination reaction from arginine. This reaction actually very commonly changes the charge state of the peptide. And so if you're looking for arginine modifications, I would caution you to be very, very careful when you're in positive ion mode. Okay, so let me show you what real HRPF data looks like so that you can get an idea of what I'm talking about when I talk about this here. So this is data from an actual FPOP experiment that we ran in my lab. Shown here on the top, this is an extracted ion chromatogram of this peptide here in the +2 charge state. And you can see we've got a nice looking chromatographic peak. If I then look for the addition of one oxygen atom, which is the most common, generally the most common oxidation product, you can see that I'm seeing multiple peaks here that elute at that mass. If I look for the addition of 2 oxygen atoms, you can see again, I've got multiple peaks that are eluting. And if I look for the loss of formaldehyde, so the decarboxylation of one of these aspartic acids here, you can see that I've got pretty much one peak here and then a bunch of noise in the background. Okay, so let's take a look at this, and I'm going to take you through what I see when I look at this particular data. So the first thing I want to point out is the retention time of your unoxidized peptide is really key when you're trying to interpret your data. And it's key for several reasons. It allows you to do several very important things when it comes to the analysis and interpretation of your LCMS data. The first thing it can do, most peptide oxidation products, when you look at them through fairly standard reverse phase chromatography that we all know and love, we're all very comfortable with it. Most of these oxidation products are going to elute immediately prior to the elution time of the unoxidized peptide. So this is going to be the area where you tend to see most of your oxidation products, is right before the elution time of your unoxidized peptide. And the exact elution time of the unoxidized peptide also gives you some very, very important information. And the reason why is because you have to remember when you're doing LCMS by electrospray, your electrospray source is basically an electrochemical cell. You can do electrochemical oxidation of peptides and proteins in your electrospray source that have absolutely nothing to do with the hydroxyl radical protein footprinting that you're trying to get information from. Okay, it's an artifact. It's artifactual oxidation that occurs during ionization. And chances are, if you've looked for it just in your standard proteomics, you've seen it from time to time. And so this happens as well in hydroxyl radical protein footprinting experiments. And it's one of several reasons why, if you're doing hydroxyl radical protein footprinting, it's always essential to run an unirradiated control. So in FPOP, since we use lasers to irradiate, I call this the no laser control. But if you're using X-rays, you can call it the no X-ray control. If you're using a Fox system, you can call it the no flash control. It's just a control where you have not intentionally generated hydroxyl radicals, but you've treated the sample the exact same in every other way. So this is that same extracted ion chromatogram of the same peptide in a unirradiated control, a no laser control. And what you can see is I get the same nice peak from my unoxidized peptide, but if I look at the addition of oxygen products, even though I haven't intentionally added any hydroxyl radicals, I'm still seeing some of these products. If I look at the plus 32, the plus 2 oxygens, some of these are actually very intense. So what's going on here? Well, one hint that this gives me when I look at it is all of these oxidation products that are eluting almost perfectly with my unoxidized peptide, these are almost always in-source oxidation products, okay? If they're present in your no laser control and they're eluting at the same time as your unoxidized peptide, these are almost always source generated artifactual oxidation products. They can happen because your source voltage is too high or because your emitter is positioned a little bit too close to your counter electrode. But regardless, they're not hydroxyl radical protein footprinting products. They're artifacts. And so you don't want to include them in your data analysis. Okay. You either want to rerun your experiment so that you don't see them, or if they're there, but they're not too intense, you want to just subtract them out of your data. So that's what these particular oxidation products are. But you still have these peaks here. Okay. Some of which, like I said, are pretty intense. These oxidation products that show up in your unirradiated or unilluminated control, but don't perfectly coelute with your unoxidized peptides, these are usually oxidation products that either existed in your protein due to protein purification and storage, or they occurred during your sample workup before the hydroxyl radical protein footprinting process. So if you're doing FPOP, these may have been oxidations that were caused directly by the hydrogen peroxide you used. Or again, they could occur due to protein storage. But either way, they have nothing to do with the hydroxyl radical protein footprinting experiment itself. And so you need to make sure that you don't include these in your calculations of oxidation for that peptide, because they have nothing to do with the protein structure. Okay, they're artifacts. So you need to either fix your experiment so that these don't exist, or subtract them out of your data. Okay. So let's look at a sample that looks a little bit nicer. So in this sample, I don't have the in-source oxidation. You can see I don't have a great big peak right here that's coeluting, and I've got my oxidation product. So this data looks a lot better. This is a lot more like what you want your data to look like. You can see most of my oxidation products are eluting just before the unmodified peptide. Some of them elute a little bit after, but they don't tend to elute far, far away. And you can see, even though this is just one mass, so this is the addition of one oxygen to the +3 charge state of this peptide, I've got lots of peaks. I'm seeing lots of different peaks here of different intensities, some of which I can clearly resolve, like this one at 14.6 minutes, and some of which I can only partially resolve, like this one here at 13.16 and its neighbor. What you need to make sure you understand is each peak represents a different reaction product. It's a different peptide oxidation product. Okay, so it has the same mass, so they're isomeric peptide oxidation products, but they're structurally different. Usually, these differences are oxidation of different amino acids. So this might be oxidation of one amino acid. This is oxidation of a different amino acid in the same sequence. They give products that have the same mass, but they're different products because the oxygen attacked a different amino acid. However, in some cases, they can actually be positional isomers, where the oxygen just attacked different regions of the same amino acid. And you'll see that sometimes, especially with things like aromatic side chains or some of the longer chain aliphatic side chains, where if you do really, really good chromatography, you can actually start to separate some of these isomers where you have oxygen addition at different parts of the same side chain, because technically speaking, they are different products. In a few cases, you can even start to pull apart stereoisomers. And this is most easily seen if you're looking at methionine oxidation products, where you have the methionine sulfoxide. that can be in one of two stereoisomers. And if you do really careful chromatography, sometimes you can start to pull apart those stereoisomers. So how do we actually go in and figure out what each of these are? Because it's also possible, remember, that some of these are not our peptide oxidation products at all. They're just other things in the mixture that happen to be isobaric. They happen to have the same or very, very similar mass to charge ratios. So how do we figure that out? It's nice if you can get MSMS data on these peptide oxidation products. It's always very helpful. The problem is that this extracted ion chromatogram I showed you here, these are all scaled relative to the base peak in the chromatogram. If I scale them relative to the unoxidized peptide, this is what you're looking at. This is your unoxidized peptide intensity, and these are your oxidation products down here. All right. Now they're real. I showed you in the previous one, they have pretty good peak shape. And if you go in and do MSMS of them, they are real oxidation products. But for most peptides, even peptides that are pretty moderately oxidized in a hydroxyl radical protein footprinting experiment, each oxidation product is going to have a very low signal intensity compared to the unmodified peptide. And this is just broadly true in hydroxyl radical protein footprinting. Sometimes you'll get a peptide where that has one really, really reactive residue to where you'll get a lot of oxidation of that one residue, and it'll be easy to assign and easy to see, but this is far more common. You've got a low amount of oxidation split among multiple different oxidation products. So the absolute intensity of any oxidation product is low. This means if you're going to use standard proteomics methods to try to design your LCM SMS experiment, so you're going to use your regular data dependent acquisition method, you're going to end up missing a lot of these oxidation products because the intensity is too low for your MSMS to switch. And even if it does switch, if you only take a single or two MSMS spectra, in a lot of cases, you're not going to be able to confidently assign what these oxidation products are. Now, using an inclusion list to where you're specifically targeting oxidation products of the peptides that you know exist can definitely help. And while it's a little bit more labor intensive to make these custom inclusion lists to get your MSMS data, it's definitely worthwhile if you have a project where you're really wanting to get in and identify all of your peptide oxidation products by MSMS. I would tell you though, if you are using inclusion lists, be careful that you go in and take a close look at your automatic thresholding for triggering on data dependent acquisition, because in a lot of cases, if you have it set to where you normally run standard proteomics experiments, some of these peptide oxidation products are going to be below that automatic thresholding limit. So even if you have it in an inclusion list, it's not always going to switch. The other thing you want to keep in mind is that unlike in standard proteomics experiments where you want to set your data dependent acquisition to only look at a particular mass to charge, one or two times and then move on to something else, so you get more depth of coverage. Remember that you've got lots of isomers, each of which has a different structure. And if you want to interrogate all of these isomers, you can't tell it to only look at or only take two MSMS spectra and then never switch again, because then you'll take a look at this first structure and you'll ignore all of the rest of them. So you have to make sure that if you're going to go in and try to do MSMS of these peptide oxidation products, you have to make sure you're designing your experiment with this kind of unique distribution of modification products that's unique to protein footprinting. You have to design your experiment with that in mind. Okay, so in our lab, what we'll actually do if we're really wanting to get structural interrogation is sometimes we'll go in and we will just we'll do an initial experiment and we'll measure that all of our +1 oxygen products of this particular peptide elute from between 12.5 and 16.2 minutes. And then we'll just go in and tell our mass spectrometer, okay, between 12.5 and 16.2 minutes, you're going to do MSMS of this mass to charge ratio, once every 2 seconds. And we'll collect data that way so that we can thoroughly and evenly cover all of these peptide oxidation products. So it's very labor intensive, but if you really want to get good MSMS, that's the kind of thing you have to do on the experimental side in order to get the MSMS data you want. But if all you want is a peptide level fingerprint, that is not actually necessary. You don't have to do that. And the reason why you don't have to do that is because we have other information that we can use along with high mass accuracy MS data that can help us get more confident identification of peptide oxidation products, even if we can't tell exactly which amino acid in the peptide was modified. The most powerful of these actually is the relative retention time of the oxidation products. So I mentioned before that oxidation products almost always elute either very, very shortly before the unmodified peptide or immediately after. And this is true, and it's true for biophysical reasons, right? Oxidation most commonly adds a polar group to a nonpolar bond. So it'll convert a benzene ring and it'll add an OH group to it, or it will take an aliphatic group and it'll add an OH group to it. So it'll add polarity to nonpolar groups, which reduces the retention time in reverse phase chromatography. But it's only adding 1 or 2 or 3 polar bonds to the same base structure. It doesn't reduce it that much. So it almost never grossly changes the retention time. It just moves it a little bit. So if it's not practical for you to do MSMS of oxidation products, then what you can do is you can use the retention time of the unmodified peptide as an anchor. And you can use that to inform yourself as far as which of all of these signals are potentially oxidation products. For example, all of this stuff here is potentially oxidation products. This is all potentially oxidation products. This is potentially oxidation products. But this stuff here at 18 minutes, this stuff here at 20 minutes, this stuff here at 22 minutes, this is not, these are not potential oxidation products. Their retention times are too far shifted and they're much more hydrophobic. So these are not potential oxidation products. If we had signal out here, we'd know it's too far shifted. This is not a potential oxidation product of this peptide. So you can use the relative retention time to inform yourself as far as which of these signals that you're seeing could be the oxidation products you're looking for. But that's not enough, because even if you do that, you're going to get things in your extracted ion chromatogram that are real, and you're going to get things that are not real, okay? Because there are lots of factors that can cause interference. You can have oxidation products of other peptides that you haven't thought about, or again, because you're really digging low in the signal, chemical noise starts to become a real issue, especially with some of these lower level oxidation products like this peak here at 14 minutes. Chances are this is a real peak. This is a real oxidation product. But once you start trying to look for things at this level of intensity, chemical noise starts to become a real issue. And if you can't do MSMS on this, you've got to use multiple lines of evidence. And so in addition to the relative retention time, in addition to the accurate mass, you want to do things like take a look at the isotopic packet that you have here. So this is the MS of the analyte eluting here at peak A. And you can see the monoisotopic peak matches very well with theoretical mass. The charge state matches with what we'd expect the charge state to look like. This looks like it's very well what we're looking for. Whereas if we look at this, this analyte eluting at 13.16 minutes here at peak B, you can see while we have signal here at 417.21, this is obviously not the monoisotopic peak for this isotopic packet. This is something else. So looking at this kind of information in your MS can help you determine which of these peaks are real and should be included in your calculations as far as calculating the intensity of your oxidation products, and which of these peaks are not real, which of these peaks are actually some other isobaric interference. Once you've gone in and looked at your MS data and you've determined what's real and what isn't, then it's just a simple matter of integration of your MS intensity, of your unoxidized peak and your various peptide oxidation products. And so I've shown you that here. This is just using Qual Browser and Excalibur to do the peak integration, but you can use whatever software you like. I already showed you why we determined this was not an actual oxidation product. I used that same method to go in and look at all of these other peaks. Most of them were. A few of them, like this low intensity peak here, was not real. But most of this other stuff did look like actual real oxidation products once you went in and looked at the isotopic signature, the charge state, and everything else. You go in and you measure the area of each peak, and then you quantify the amount of oxidation. And there are two ways that are commonly used in the literature to quantify. If most of your signal is either unoxidized or it's one oxidation event, and by oxidation event I mean the addition of one oxygen atom or a single loss of formaldehyde or a single carbonyl. So you had one event that happened to the peptide that caused the oxidation product, then you can simply calculate a percent oxidation, where you sum the intensities of all of your oxidation products, and then you divide that by the sum of the intensity of all of your oxidation products plus the intensity of the unoxidized peptide. If, on the other hand, you have a large amount of signal that required multiple events to make that signal, so for example, the addition of 2 oxygen atoms or a plus 16 and a plus 14 occurring on the same peptide to give you a total mass shift of plus 30, or a plus 16 and a minus 30 happening on the same peptide. So signal that happened where you had 2 different hydroxyl radicals attacking the same peptide and forming 2 different oxidation events that are independent of each other, then percent oxidation is actually going to underestimate the amount of oxidation that you have. And you want to move to a bit more complicated formula that gives you the average oxidation events per peptide. And it's very, very similar. The only difference is in the numerator, you're going to weight the intensity of each oxidation product multiplied by the number of oxidation events it took to make that product. Okay, so oxidation products that take a single oxidation event to make, in this case, I exemplify that with a plus 16, are weighted by 1. Oxidation products that take 2 separate events, so a plus 32 or a plus 30 or a minus 14, will be weighted by 2, by the number of oxidation events it took to make that product, and so on and so forth. Again, divided by the sum total of all of the summed intensities of both the unoxidized and all of the oxidized versions of that peptide. Now, both of these methods of quantification assumes that the ionization efficiency of the unoxidized peptide and the ionization efficiency of all of the peptide oxidation products are the same. And in general, this is pretty true. Of course, there is some fudging in this assumption. The ionization efficiency is not the exact same, but for most oxidation products, it's really close. Again, there is one major exception in positive ion mode, and that is the deguanidination product of arginine. That changes the ionization efficiency of the peptide by so much that most experts actually recommend that you don't try to quantify arginine deguanidination in positive ion mode. If you really want to quantify arginine deguanidination, you probably want to be looking in negative ion mode. And always, always, always subtract your no laser control, because most samples are going to have some level of artifactual oxidation, and you need to subtract that from your peptides if you want to get a true measurement of how much HRPF related oxidation you have of that peptide. Now, HRPF data, like all data, has error. Where does the error in HRPF data most commonly come from? So there are 2 types of error that are common. There's systematic error and there's random error. Systematic error is most often caused due to incorrect peak assignment, either due to you mistakenly count a artifactual oxidation as an HRPF related oxidation, so you don't properly subtract your background, or more commonly, you mistakenly identify something that's not a peptide oxidation product as a peptide oxidation product. And so these systematic errors are things that I've already tried to teach you how to avoid as much as possible. What's more common is random error. And we actually did a pretty close study looking at random error and what causes random error in HRPF experiments. And what we found is that random error is most commonly caused due to low peptide intensity and the influence of chemical noise on this low intensity signal. As your peptide gets, or as your peptide oxidation products get higher in intensity and total intensity, the amount of error that you get for that peptide oxidation product decreases significantly. And so in a lot of cases, if you end up getting HRPF data that looks like this, what's here in solid yellow with these big error bars, right? In a lot of cases, the first thing I recommend trying to do to fix it is just inject more of your sample on column. So you can boost up your total signal intensity. And that's exactly what we did in this experiment here. Shown in yellow is a HRPF sample where we just did a 2 microliter injection of our digest. And then shown in white, we used the exact same instrument method, the exact same instrument. Everything was the exact same sample. Just instead of injecting 2 microliters onto the column, we injected 5 microliters onto the exact same column. And you can see our error increased in a lot of cases very significantly, even though there was no significant differences in the mean for any of these measurements. So if you're seeing big error bars in your HRPF data, the first thing I recommend is just put more sample in your mass spectrometer, increase that signal intensity so that you can dampen down the influence of the chemical noise that it has on these low intensity oxidation products. Okay, once you have that, once you've got your results, you've got your percent oxidation or your average oxidation events per peptide, then the question becomes, what are the statistics that we normally use? How do we know if some change that we see is real? And the statistics you use depends on what hypothesis you want to test. So there are a couple of approaches that are pretty common in HRPF. The most common one by far is, you've got one reference sample and you've got one test sample. So you've got your protein in 2 different structural conditions, and you want to know what is changing. So you want to look at each region of the protein independently and tell, did this region change in sample A, in condition A versus condition B? Yes, no. Okay, did this region change in condition A versus condition B? So the way that that's most commonly done is using just a standard two-tailed Student's unpaired t-test. With whatever p-value that you're comfortable with. The default is 0.05, but it really depends on what you're comfortable with. However, there is an application for HRPF that's becoming a bit more popular, and that a lot of biopharma people are interested in. And that is trying to look at similarity and say, did anything change? Okay, so I'm wanting to change the formulation of this drug, and I want to know, does the formulation change anything? And in that case, typically what we do 2-tailed unpaired t-tests for each peptide, but once you do that, you need to make a correction because you're doing multiple comparisons. Okay, and so if you don't do a correction for multiple comparisons, you're going to find changes by random chance much more often than if you were just looking at one peptide. So you need to do a correction for multiple comparisons. There are a couple ways you can do that. You can do a CDAC correction post hoc, or you can convert all of your data to z-scores and perform two-way ANOVA in order to see if there's any change. But this is a bit more detailed. Most commonly, though, what most people are doing, just looking at one reference sample and one test sample, is just standard two-tailed unpaired Student's t-test. And once you have your data, once you have your statistics, there are a few ways that are commonly used to look at your HRPF data so you can understand what's going on. You can always tabulate your data to where you just have a table of, okay, this is the oxidation in condition A, condition B, standard deviations, fold changes. Tabulation is very thorough. A lot of journals really like that. A lot of reports really like that, but it's also very dense and it's hard to really look at and get at a glance an idea of what's going on. So if you want to visualize the data outside of tabulation, there are 2 ways that are commonly used in the literature and in presentations. Probably the most popular way is just the standard histogram. In the histogram, you've got condition A as one bar and condition B as another bar. Standard deviations are plotted. And this is really good at showing both the absolute magnitude of oxidation of each amino acid. So you can see which amino acids are oxidized poorly and which amino acids are oxidized at a high level, and showing the magnitude of change between condition A and condition B, because you can look at the differences between the two conditions for any given peptide. What it's not good at is it's not good at showing statistical confidence. So when you're looking at a histogram, usually what you'll do is you'll apply a particular p-value, and then you'll just mark all of the peptides that met that p-value. But it's not good at showing a level of statistical confidence. Okay, so something that was significant at a p-value of 0.049 is going to look the same as something that was significant at a p-value of 0.0001. And there's not a good way in a histogram to show that. The other way that's been used a bit more recently is the volcano plot, which a lot of people are much more comfortable with when it comes to things like proteomics. Volcano plot is very good at showing the relative magnitude of change. So along the x-axis, you have a log2 of the fold change between your reference versus your test. And then it's also very good at showing statistical confidence, because along your y-axis, you have the negative log 10 of your p-value. So the more statistically confident you are that the change is real, the higher it is. And then the greater the change in terms of fold change, the more to the left or the right it is. So this is really good at looking at magnitude of change and statistical confidence, but what it's bad at is it does not show you at all what the absolute magnitude was of oxidation. So you can't tell which peptides were heavily oxidized versus which were lightly oxidized. So it really depends on what you're wanting to show and what you're wanting to emphasize, but you'll see both of these in the literature. Okay, so a lot of people at this point will stand up and say, you know, this seems really, really hard. And I'm not going to lie to you, it is the slowest part of FPOP and HRPF in general. The experiment, it takes you maybe an hour to irradiate all your samples. You start the digestion, you run it overnight, you put it on your LCMS, you can be done with the experiment in a day and a half. But the data by itself can take you a week, maybe more, to analyze, depending on how many peptides you're looking at and how comfortable you are looking at the data. But with that, it does get easier with practice. So something that would take somebody 2 weeks to do when they're first starting, then will take them, 2 hours to do once they're comfortable looking at the data and they know they have their workflow down. You get to where you can look at these oxidation products and much more quickly determine which ones are real, which ones are not. It's really important that you have good replicates. Because the more you look at data that look the same, the faster you can determine which of these oxidation products have I seen before, which ones do I already know are real, versus which ones have I not seen before, which ones need a little bit more of my time to look at. So the more you analyze the same protein or the same sample or the same type of experiment, the easier it gets every time you do it. The hardest thing by far to assign are the low intensity oxidation products, because when you get to low intensity oxidation products, your C13 isotopologs are near the chemical noise level. Your signal intensity tends to be unstable, again, because you're near that chemical noise level. It's really, really tough. The good news is that mistakes on low intensity peaks tend to be small mistakes. Okay, so if you're looking at a peak that's really, really low in intensity, where the area of that peak is really, really small compared to everything else you're looking at, you might make a mistake on it. Chances are that mistake is going to be a pretty small mistake in the overall scheme of things, just because low intensity peaks are low intensity. They tend not to add that much to the signal. So once you have your data, what do you do with your data? How do I turn this histogram into knowledge, right? How do I turn this into structural knowledge? And when it comes to that, footprinting, much like a lot of other structural techniques, they can really benefit from a model. Whether it's a good model, whether it's a so-so model, a model gives you something to test. It gives you a hypothesis to test. It gives you some way to contextualize your data. So whenever you're visualizing HRPF data and trying to interpret it, models are very, very helpful. And plotting your data on a structural model really helps you interpret it. When you're plotting data on a structural model, the key factor you want to plot is fold change, okay? It's not absolute magnitude of change. Absolute magnitude of change can vary significantly depending on what the amino acid identity is, whether you're oxidizing a tryptophan versus whether you're oxidizing an asparagine. You can have a very um, big difference in absolute oxidation that reflects very similar differences in structural change, in the change in the solvent exposure. So what you really want to look at is fold change. And what I usually like to do is I like to plot fold change on my structural model. And I like to use a color code that looks kind of like this, to where things that are protected that show a twofold or greater reduction in oxidation, I plot as blue. Things that show significant reduction in oxidation, I will plot as cyan. Things that I see are oxidized but show no change, I'll plot as white or gray, depending on what I'm doing. Things that are exposed less than twofold, so it showed a less than twofold increase in oxidation, I'll plot as magenta. And then things that show a greater than twofold change in increase in oxidation, I'll plot as red. And so this gives me a nice, easy way to take a look at things and figure out what exactly is going on. There are two common ways that people will plot this. The cartoon diagram shown here as ribbon is very popular because this is how people like to look at protein structures, right? This is what people are comfortable with when they look at protein structures. But one thing to keep in mind is, remember always that HRPF at its heart is looking at protein surfaces. So in a lot of cases, it's really useful to do a surface representation of the protein model when you're trying to understand what's going on with the data. So in this case, we had a protein that has a very large ligand that binds to it. So it's a very large linear ligand that binds to it. We were trying to understand, where does the ligand bind and what does it do? And it turns out the ligand actually binds in this groove right here, which we identified both sides of the groove. And then when it binds, it appears to actually cause this these two domains to open up, to kind of open that groove up. And you can actually see two amino acid, or you can see two regions on opposite domains that become more exposed when this cleft opens up. So if I just looked at the data like this, I wouldn't be able to tell any of that, right? I'd be able to tell, okay, this region appears to be involved because lots of parts of it are protected. This region doesn't appear to be involved much, but other than that, I can't tell a whole lot. But when I put it in the context of a model, now I can actually tell what's going on. So having a model that you can actually use to visualize your data is very helpful. Okay, so I've walked you through how to do this manually, and now I want to try to talk to you a little bit about some software options that are available. The first thing I want to tell you is that the software that people are really comfortable with for identifying post-translational modifications, they usually struggle with quantifying HRPF oxidation products, because they're usually designed to look at relatively few possible modifications per peptide. So things like, phosphorylation, you're looking at a couple of different sites per peptide, whereas with HRPF, you've got every amino acid that can be oxidized. And in a lot of cases, they are. So the software that specializes in more standard PTMs usually don't do a great job of identifying what we're looking at. They like to see well-behaved XIC peaks that look pretty symmetrical, and you don't get that a lot with oxidation products. And they really don't like trying to identify chimeric MSMS spectra. And in HRPF data, a lot of times you'll have two different oxidation products that partially overlap, or even completely overlap in the chromatogram, to where what you end up fragmenting is a mixture of multiple structures. And most software really hates that. Protein Metrics actually has put out some software related to Bionic that does address HRPF data, and it does much better than standard PTM-based traditional proteomics software. I have found in my personal lab, using this software, we always have to go in and validate quantification, because our manual interpretation will almost always give us different answers than Protein Metrics does. But Protein Metrics, their software has been a good first pass that we've used in the past to try to help us get started in the manual process. GenX has built a product called Foxware that's actually been built to work with data without having thorough MSMS of oxidation products. And it actually works through automating the exact same manual steps that I just went through with you. It focuses on relative quantification. It does not try to exactly determine the site of oxidation by MSMS. But if you're looking for peptide level quantification, this is a product that I'm really proud of and that I use in my own lab. And this is just giving you a brief overview of what it does. Again, it goes through the exact same steps that we went through manually. It converts your vendor LCMS data to an MZML format. It finds the unmodified peptide retention times based on MSMS of the unmodified peptide. It goes through and automatically calculates masses for all of the oxidation products of interest. It will extract the extracted ion chromatogram for all of those masses. And then it will go in and look at every peak that falls within that retention time window relative to the unmodified peptide and determine for each peak that it finds in that extracted ion chromatogram, is it a real oxidation product? And that's based either on automated feature detection, which is a fairly conservative method for identifying, or if the automatic feature detection does not identify it, it will go in and it will look at charge, the M+1 isotopolog to make sure that it looks like an actual organic molecule, and the lack of an M- 1 isotopolog to demonstrate that we are looking at the monoisotopic peak. It will subtract your unirradiated controls automatically and calculate your average peptide oxidations. And so this is just taking a look at the software itself real quick. I don't want to spend a whole lot of time looking at this because this is really the kind of thing where if this is something you're interested in, you really want to get a software demo rather than just have me talk about it. But it makes these extracted ion chromatograms. It uses MSMS to identify which of these peaks is the actual unmodified peptide. And then it uses that unmodified peptide to set retention time limits to look for peptide oxidation products in your chromatographic window. It'll then go in and calculate all of the different oxidation products that it's going to look for based on different oxidation products and a relative occurrence. Which ones are more common? So it'll look for more occurrences of those, and which ones are more rare? It'll look for fewer ones in the combinations that it calculates. It'll then go in and make extracted ion chromatograms within that retention time window and identify all of these and mark which peptides meet its criteria and which peaks don't meet its criteria for each of the oxidation product combinations that you gave it. And then you can go in and look at each peak individually, and you can see this is why this one met. This one was actually found as a feature here. This one was not found as a feature, but if you go in and look at the different isotopes, it meets all of our isotope criteria. And then this one was not found as a feature and did not meet our criteria because it does not have an M+1 isotopolog. And this one did not meet our criteria because it's not a feature. While it does have an M+1 isotopolog, it also has an M- 1 isotopolog, so it's probably not the monoisotopic peak. It'll then go in and make the histograms and the volcano plots, just like we talked about doing manually. It'll give you a few different options for doing that for if you need to do different kinds of troubleshooting. And then it'll actually start to plot your data onto user-defined models so you can see different levels of oxidation plotted in different colors on your structure. So that is a brief introduction of how to do HRPF data analysis and data processing. Thank you very much for your time. If you have any questions, I would be happy to answer them at this point.
Thanks, Josh. So we have one particular question that has been submitted. I'll go ahead and read it off to you. So you've described the use of the analysis basically at a peptide level. What would you do if you wanted to analyze the data for residue level resolution?
Okay, all right. So residue resolution is a bit more challenging, and there are a few ways that you can actually approach that. The most common way is to go in, and if you'll let me back up on my slides, I'll try to use some visual aids here. Is you go in and what you do is you try to get MSMS data on all of these peptide oxidation products. All right, so you try to chromatographically separate as many of the peptide oxidation products as you can and get good MS data on all of them to identify which amino acid has been modified. And then what you'll do is you'll go in and you'll take the peak area of each of those different oxidation products separately and use that to identify the percent oxidation at each amino acid. So this seems to work pretty well if you're wanting to look for changes in oxidation. So for example, if you take a look at this particular peptide in condition 1 versus condition 2, and you notice that most of these oxidation products look the same, but this particular peak has decreased from being very intense to being not very intense at all, then you can identify what amino acid is oxidized in this peak, and you can then go in and say, okay, the amino acid that changed is this particular amino acid. And that seems to work pretty well. Um, it falls down in a few different places. It's not good for doing absolute quantification. So if you want to say that the oxidation at this amino acid went from 17% to 12%, it seems to not really do well at that. On the other hand, if you want to say, this particular oxidation in this amino acid changed twofold, it seems to do a much better job of that. It does require you to have good chromatographic separation of all of your oxidation products and to have good MSMS. And I already mentioned why that's difficult. Some of these oxidation products do not separate. And if they don't separate, you end up looking at a chimeric MSMS spectrum. So you're actually fragmenting multiple structures in a single MSMS spectrum. And the resulting MSMS data are very hard to assign confidently. And so you can end up having misassignments that way. And in some cases, some of these oxidation products are just low enough intensity to where it's hard to get good MSMS signal out of it. So it is challenging. The other way that has been used is that you can actually go in and do electron transfer dissociation of all of these peaks. So if you do an electron transfer dissociation fragmentation, let's say you do one every 10th of a second, to where you get full coverage of all of these peaks. Then what you can do is you can go in, you can average all of those spectra together, and then you can use the change in the intensity of each fragment pair. So you've got a C2 ion, and then you've got a C2 ion plus oxygen. And you can take the relative intensity of that C2 ion and that C2 ion plus oxygen and determine how much of that C2 ion is oxidized. And then you can do the same for the C3 ion and see how much that changed from the C2 ion and assign that oxidation to the third amino acid, and so on and so forth going down the line. That method is better at doing absolute oxidation quantification in my research experience, but it's much more labor intensive, and getting high quality ETD spectra is even harder than getting high quality HCD or CID spectra. So it's really, really challenging. But just as a general strategy, that's how you want to do it, is you really need to at least identify what each of these oxidation products are in order to start doing residue level quantification.
Great, thanks. Another question that was submitted, in terms of data processing, are there any specific requirements or nuances for FPOP versus maybe X-ray synchrotron radiation or even the flash oxidation?
Right. So the FPOP and the flash oxidation are basically identical. The only difference between those two are you using a laser or are you using a flash lamp? So those two are treated about the same. X-ray is a little different because of the way the X-ray experiment is done. In the X-ray experiment, they're actually doing multiple experiments generally. So what they'll do is they will oxidize the same sample at different time points, so for different periods of time, to get a gradually increasing amount of oxidation from sample to sample. And then what they want to do is they want to measure basically the slope of that increase to get a rate constant. And then once you have that rate constant, you look at the differences in the rate constants. That part is a little bit different. There's no reason why you couldn't do X-ray similar to how you do FPOP with a single time point and a histogram-based analysis. That's just not what's commonly done. The way that they do it is considerably more labor intensive. Strictly speaking, it is also more rigorous. So, and you can do that. You can do that experiment, that rate constant experiment. You can do that with FPOP. You can do that with Fox. Just for most applications, people don't do it because they don't feel that it's worth the extra sample and worth the extra time. So it really depends. Overall, the data analysis between the two are very, very similar. I would almost say identical.
Okay, great. And I think we have time for one last question. So here it is. So in terms of the data processing, is there anything that you could particularly or practically perform, any test you can practically perform to ensure that you're not seeing any artifactual outcome, for example, a change in conformation?
So it depends on all right. So it depends on what you mean by artifactual outcome. So if you're talking about a change in conformation due to the labeling itself, then yes, there is absolutely a test you can do for that. And that is part of why the X-ray synchrotron people do that kinetic analysis, where they'll oxidize a little bit more and a little bit more and a little bit more. And that's because if you get an oxidation-induced conformational change, what will happen is that relationship, instead of being giving you a linear slope, it'll deviate from linearity significantly as the protein starts to change its conformation at higher levels of oxidation. Um, Similar methods have been used in FPOP in the past. The Fox system uses a similar method, basically to calibrate its measurements. For the first time it works with a new system. You'll do oxidation at different laser intensities or different flash intensities. So you'll make more hydroxyl radicals, and you'll measure how that level of hydroxyl radical is related to the amount of oxidation of each peptide. And they should be linear or very close to it, with a few exceptions as you start to saturate sites of oxidation. And yeah, so that's probably the most common way. There are other ways that are a little bit technically more challenging. So you can look at the intact protein itself instead of looking at individual peptides. You can look at the intact protein and the protein oxidation products and measure the distribution of oxidation products based on intensity. That's the way that my lab worked with the Gross lab to initially prove that FPOP didn't change the conformation of protein during labeling, was by looking at that distribution of oxidation products. But now I'd say the most common way is basically to expose the protein to different doses of hydroxyl radical, and then look and see if that exposure is linear, with a few exceptions as you start to saturate certain sites of oxidation.
Great. Thanks, Josh. So I think we're pretty much done with the Q&A at this point. What I'd like to do then is thank everybody who attended today. And just to remind you all that this video, this session will be available for on-demand viewing probably sometime next week. You will get an email from us prompting you to that effect, and you can view the video at our website at genxtech.com. Also, I want to remind you all that the next installment, or the final installment for this 5-part series, will be presented next year by Professor Lisa Jones on the 10th of February. And if you happen to be interested in FPOP instrumentation, data processing software, or if any contract services, please go ahead and reach out to us and drop us a line at info@genxtech.com. And both GenX Technologies and Neoproteomics would like to wish you a very happy, and during this troubling time, of course, a very healthy holiday season. And thanks so much. And we'll talk to you all next year. Bye now.